Good morning and welcome all. We welcome you all in the ISFCP Dialogue Series under the aegis of IQAC IIC ISFCP, organized by ISF College of Pharmacy and Autonomous College Moga, Punjab. Today's special guest and speaker, Professor Dr. Pooja Chawla, and the title of his talk is Role of Analytical Techniques in Pharmaceutical Sciences. We welcome Dr. Pooja Chawla. Please proceed.
Thank you, Dr. Siddharth Mehan. Thanks a lot for giving me this opportunity. As Dr. Siddharth has mentioned, the title of my topic for today's lecture is Role of Analytical Techniques in Pharmaceutical Sciences. As we all know that being in the pharmaceutical sciences, that the importance of pharmaceutical analysis is a big deal. What is pharmaceutical analysis? It is defined as the application of analytical procedures used to determine the purity, safety, and quality of drugs and chemicals. Pharmaceutical analysis, it's a branch of pure chemistry that involves identifying substances or drugs, purification, separation, quantification, determining the structures of chemical compounds used in the making of a pharmaceutical product and various pharmaceutical samples. So, if I go to the technology overview, there are various instruments that have revolutionized chemical analysis. It replaced wet chemistry methods, provides higher sensitivity and accuracy, increases lab productivity, and reduces manpower due to automation, minimizes operator skill requirements and errors. Coming to the advanced technology, there are a number of techniques like separation techniques, high performance liquid chromatography, gas chromatography, mass spectrometry, LCMS, GCMS, MALDI, TOF, life science instrumentation, sequencing, DNA synthesizers, organic and peptide synthesis, and electrophoresis. Spectroscopic techniques. Coming to the advanced technology instruments, there are various spectroscopic techniques that include atomic absorption, UV visible, ICP-MS, fluorescence, and near infrared. Surface sciences include electron microscopes, light microscopes, scanning, etc. Material characterization like thermal analysis, viscometer, and lab automation, including information management, robotics, liquid handling, and microplate reading. They are analytical instruments. They have revolutionized the demand by industry. Like, as you can see in this slide, there are 4% electronics, 4% metals, 4% polymers, 3% oils and gases, 5% chemicals, then followed by 6% hospital, agriculture, biotech. And here you can see our pharma industry, we include 19%, we consume 19% of the analytical instruments. In academics, as we are from the academia, 19% equal contribution to the industry. 19% analytical instruments are utilized in academia and 12% by government institutes and organizations. Now, coming to the demand by function in the pharma industry, here you can see analytical services, they consume 24%, QA/QC 20%, method development followed by R&D that consumes 43% of the demand of the instrumentation, and some other fields they are 6%. Now, a big question comes to mind that why pharmaceutical analysis? Because I am from the pharmaceutical chemistry background, and by other departments or other branches like pharmacology, pharma analysis, QA, QC, other departments, they think that why pharmaceutical analysis is important for us. Here you can see the pharmaceutical analytical techniques, pharmaceutics, pharmaceutical chemistry, as I've already mentioned, pharmaceutical analysis, pharmacology, quality assurance, pharmacognosy. I mean to say every branch includes the importance and utilization of pharmaceutical analysis, because here you can see that in case of pharmaceutical chemistry, if we synthesize a chemical moiety, let us say thiazolidinone derivatives or any other chemical salt, it has to be analyzed by two methods: physiochemical parameters and spectral parameters. If we have to confirm the structure of the compound that is being synthesized, first of all, we have to go for its UV. after the physicochemical parameters, we have to go for its UV, then confirm the what you call functional groups, that is with the help of IR spectroscopy, and followed by the confirmation of the structure by mass spectrometry, that will tell about the molecular formula of the compound, and the final full and final structure is confirmed on the basis of nuclear magnetic resonance spectroscopy, that tells about what type of and how many protons and carbons are present in the particular chemical entity that we have synthesized in the lab. Now, coming to the importance, first of all, if I don't talk about the pharmaceutical chemistry, they identify the identification of the drug in the formulated product. This is the importance for pharmaceutics department. Then determination of active ingredient or additional impurities, stability of the drug is carried out, rate of drug release from its formulation inside the body, so as to calculate the pharmacokinetics and the availability of the drugs inside the body. The quantity and purity of pure drug that meet specification, concentrations of specified impurities which may be present in the sample, concentrations of the drug in plasma or biological fluids, determine the pKa values, partition coefficients, solubilities, and stability of the drug under the development. Here you can see there are various analytical techniques which are used in the simultaneous estimation. First of all, we can go for spectrometric techniques like UV visible, IR, mass, and NMR that I have already told you. Then we can go for the chromatography techniques that actually what they do is the separation and purification of the compounds, which may include HPLC, gas chromatography, TLC, HPTLC, and UPLC. There are, as the technology advances, we have got a number of hyphenated techniques. Here you can merge LCMS, GCMS, ICP-MS. LCMS, for example, if the compound is purified or a natural product is extracted from the natural source, then after the purification, we can determine actually the molecular mass of that particular compound. So after the chromatography, the compound, the molecule can directly get into the mass spectrometer and we can determine its molecular mass. Now, coming to the spectroscopy, what is spectroscopy? It is basically the measurement and interpretation of electromagnetic radiation absorbed, scattered, or emitted by the atoms, molecules, or other chemical species. This absorption or emission results in the change in the energy states of the interacting chemical species. See, the spectroscopy, it is nothing but it is the game of the electrons. If I'm talking about the UV, because if any electromagnetic radiation, I will tell in the next forthcoming slides, that if any electromagnetic radiation fall on the sample, so there are, as we know, that the molecule is made up of atoms, which in turn are made up of electrons, protons, and neutrons. So electrons being in the outermost shell, they get excited, and the complete story or the journey of the electron in its excitation form tells about the technique, that is the spectroscopy. So in case of UV radiation and electronic excitations, the difference in energy between the molecular bonding and non-bonding and anti-bonding orbital ranges from 125 to 650 kilojoules per mole. The energy corresponds to electromagnetic radiations in the UV region, that is 100 to 350 nanometer, and visible region, that is 350 to 700 nanometer of the spectrum. In some books it is written 200 to 400 in the UV and 400 to 800 in the visible region. And for comparison, the EM spectrum, it is shown ahead. Here you can see that the source of the radiation, it passes through the entrance slit, and through entrance slit it goes into the, here you can see this is the monochromatic part of the assembly. Monochromator, mono means one, chromato means color or wavelength. The function of the monochromator is the cutting short of the polychromatic light into the single wavelength, that is monochromatic light. And when it falls to the sample, the electronic excitation takes place, and after that those transitions get detected by the detector. Here in this you can see the whole spectrum of the UV and visible, or I can say the electromagnetic radiations, from gamma, X-rays, ultraviolet, ultraviolet, and in between the ultraviolet and infrared, there is visible region, what we call as VIBGYOR. Below the violet is the ultraviolet in terms of nanometer or in terms of the wavelength, and above red is the infrared. Now, it is based upon the spectroscopy, it is based upon absorption and emission. Absorption, if a molecule absorbs some radiation, either it will keep it up to itself only, or the in turn the radiation that are absorbed, they will be emitted out. So this is the basic principle: absorption and emission. Absorption, the transition from lower level to the higher level with the transition or transfer of energy from the radiation field to an absorber at a molecular solid. And in case of emission, transition from higher level to the lower level with transfer of energy from the emitter to the radiation field. And if no radiation is emitted, the transition from higher to lower level is called as the non-radiative decay. It will decay somewhere on the way. Coming to the principles of spectroscopy, the principle is based upon the measurement of spectrum of a sample containing atoms or molecules. Spectrum, it is graph of intensity of absorbed or emitted radiation by sample versus frequency or wavelength. And spectrometer, it is an instrument designed to measure the spectrum of a compound. As I have already explained the absorption spectroscopy, that when the electromagnetic radiation is absorbed, the transition from the low level to the higher state will take place. And if it is UV, the absorption will be at the 185 to 400 nanometer, visible 400 to 800 nanometer. In case of IR, it will be 0.76 to 15 micrometer range. And the more effectively molecules absorb light of a given wavelength, greater will be the absorption. At the absorption spectroscopy, it is based upon Beer-Lambert law. Beer-Lambert law states that the absorbance is directly proportional to the concentration of the sample used in the experiment. That is the Beer's law. And Lambert says the absorption is directly proportional to the length of the light path. That is also called as path length, which is equal to the width of the cuvette. So here you can see the statement: A is equal to epsilon CL. A is the absorbance, epsilon is the molar absorptivity, C is the concentration, and L is the path length. Intensity of a beam of a monochromatic light decreases exponentially with the increase in the concentration of absorbing substances. Mathematically, we can very well correlate that if everything is absorbed, nothing will be emitted out, or vice versa. So this is the Lambert-Beer law. Here you can see in the upper part, the light source monochromatic chops the light from the polychromatic to monochromatic. After that, with the help of the rotating disc, a part of the sample of the radiation will pass the sample and other will pass to the reference, that is the standard. And after passing to the mirrors, they will go to the detector, and we can detect, or we can take the readings. The intensity here in this example, you can see the intensity of beam of monochromatic light. It decreases exponentially with thickness. It decreases exponentially with the thickness as well as with the concentration of the sample. Here you can see for aspirin, it is 229 nanometer, and for salicylic acid, it is 296 nanometer. And in the lower graph, you can see the transmission versus concentration. And in the right side, you can see absorbance versus the concentration. That is the linearity study of the paracetamol. Now, there are various deviations from the Beers and Lambert law, because every molecule or every substance cannot obey that at all the concentrations. So it is valid over a wide range of concentration if the structure of the colored ion or colored non-electrolyte in dissolved state does not change with concentration. And a specific point is that the Lambert-Beer law, it is applicable for the what you call the dilute solution. For concentrated solutions, this does not come into picture. So there are various deviations from the Lambert-Beer law. So deviations may occur if the colored solute ionizes, dissociates, or associate in the solution. Benzyl alcohol in chloroform exists in the polymeric equilibrium. Here you can see four molecules of the benzyl alcohol. They associate to give the polymer that is called as the polymer of benzyl, and it is in the continuous equilibrium with the non-bonded. So another example of the deviation is the change in the color of dichromate ion on the solution. Here you can see the example of potassium dichromate. So dichromate ion is in orange in color, and when it dissociates, it will give the chromate ion that will be yellow in color. So solution of dichromate is converted to chromate, and absorption shift from 450 nanometer to 350 nanometer, and they may be the positive as well as the negative deviations. So deviation is also due to presence of some impurities that may fluoresce or absorb at the absorption wavelength. They occur if the monochromatic light is not used. Deviation may also occur if the width of the slit is not proper. They may allow the stray radiations or the undesirable radiations to fall to the detector, and they may also occur if the solution undergoes polymerization, as I have already explained, and it cannot be applied to the suspensions. So, as far as the emission spectroscopy is concerned, it is an analytical technique in which, as I have already mentioned, that which transition occurs from higher energy state to the lower energy state with transfer of energy from emitter to the radiation field. And if no radiation is emitted, then that will be your non-radiative decay. The example is the mass spectrometry. There are various interactions of the EMR with the matter. There will be electronic energy levels. At room temperature, the molecules are in the lower state, and as we know that as we provide them the energy, they will transit from the lower energy level to the higher energy level. Another one is the vibrational energy. That is, when you provide the radiation, they may get excited, but the energy is lesser so as to excite the electrons from their lower energy level to the higher energy level. So what they will do, the molecules, the atoms, they will start vibrating at their own levels or at their own place at a higher amplitude. Then the next one is the rotational energy levels, and these energy levels are quantized and discrete energy levels, and the spacing between the energy levels are even smaller than the vibrational energy levels. And here you can see the energy rotational is lesser than energy for vibrational, which is lesser than the electric for the energy for the electronic. So internal energy of all the molecules, it is a total of transitional or electronic or vibrational or rotational energy levels. So there are various transitions that take place from the electronic transitions, that is sigma to sigma star, pi to pi star, n to pi star, and n to pi star. So the main part that is in case of the UV spectroscopy, that is a chromophore. Chromo means color. So they provide color to the molecule. So they may also be called as the chromospheres. They are the functional groups that are capable of absorbing the radiations above 200 nanometer due to N to pi star or pi to pi star transitions. Examples are NO2, NOCO, for example, like that. Oxochromes, they are the functional groups. They are attached to chromophores, which modify the ability of the chromophore to absorb light. They themselves do not give the they have their own wavelength, but they impart the frequency, or they impart the absorbance intensity of the molecule. They are such type of functional groups. There are various shifts, that is bathochromic shift, hypsochromic, hyperchromic, and hypochromic. Bathochromic shift is the red shift when the after addition of any oxochrome, the wavelength goes towards the higher side. Then that is called as the red shift. If the wavelength goes towards the lower side, then it is the blue shift. If the intensity of the radiation increases, then it is the hyperchromic, and it is hyperchromic if the intensity is decreased. This is the slide that is showing the all the four shifts in single slide. So now coming to, because if I give the gist of the UV, what we can get from UV spectroscopy, that we can get the amount of conjugation. See, conjugation is the minimum thing that is required for any compound to be UV active. So this can tell us about the various type of chromophores or oxochromes which may be present in the molecule, and the extent of conjugation which is present in the molecule under study. Now coming to the infrared spectroscopy, this is the second very important part of spectroscopy. It is a technique that is conducted with an instrument called as infrared spectrometer or spectrophotometer, which produces an infrared spectrum. IR, or it is also called as the vibrational spectroscopy. It is the measurement of the interaction of the infrared radiation with the matter of absorption, emission, or reflection. It is used to study and identify chemical substances or functional groups in the solid, liquid, or gaseous form. See, this is based upon the principle that when a molecule is given the electromagnetic radiation under the IR region, the energy is there, but it is not that much higher as compared to UV that it can excite the molecules, the electrons, from lower energy level to the higher energy level. So in that case, what happens? The electrons start vibrating at their own places. These are called as vibrations. So it exploits the fact that molecules absorb frequencies that are characteristics of their structure, and they will vibrate at their particular frequency only. And these absorptions occur at resonant frequencies, that is, the frequency of the absorbed radiation matches with the particular vibrational frequency, and the energies are affected by the shape of the molecule, molecular potential energy surface, the masses of the atoms, and the associated vibrational coupling. There are different types of vibrations. One is the stretching and other is bending. Stretching, that is nothing but the change in the bond length or bond axis. It may be symmetric or asymmetric. In symmetric, the atoms, they may either go away from the central atom or they will come towards the central atom. And in case of asymmetric, the one atom is going away from the central atom and other atom is coming towards it, and vice versa. In this process, the bond length or bond axis will be changing, increasing or decreasing. In case of bending, it includes the change of the bond angle. So in case of this is again of two types: in plane bending and out of plane bending. In plane bending is further of two types: scissoring rocking, and the out of plane, that involves wagging and twisting. In case of scissoring, see, the central atom is placed over there, and the other two atoms, they will either move towards or away, just like the scissor. Like this is the central atom, these are the two other atoms. If they are moving towards the central atom or they are going away from the central atom, here you can see the bond angle is changed. That is, this process is called as the scissoring, just like the scissors. In case of rocking, the other atoms, they will either move away from the central or they will move, they will move in the same direction, just like the wipers of a car. And in case of both of these, scissoring and rocking, they are happening in the same plane. But in case of out of plane bending, one is wagging and the other is twisting. In case of wagging, one atom will be moving, both the atoms will be moving either above the plane or below the plane. Okay, that is called as wagging. And in case of twisting, the one atom that will be moving below the plane and other atom will be moving above the plane. So this is the plane, one is moving above and the other is moving below, or you can say we can twist like this. So this is, as this involves the plane, so they are called as out of plane bending. This is the flow diagram of a dispersive IR spectrometer. Here you can see the IR source. The radiation will fall the reference as well as the sample, and after passing to the chopping device, we can get the spectrum of the IR spectrum of the compound. Factors that affect the vibrational frequency in IR, they are coupled vibrations, Fermi resonance, electronic effect, and hydrogen bonding. See, in case of coupled vibration, sometimes what happens is that two vibrations, their frequency may collide or coincide with each other. In case of Fermi resonance, in spite of going from one level to the other level, vibrational level, the molecule may go far above that, and it is the resonance, it is the combination between the overtones as well as the that is called as the Fermi resonance. And in case of electronic effect and hydrogen effect, the electron donating groups or electron withdrawing groups, they will impart the effect. In case of hydrogen bonding, there are two types of hydrogen bonding. One is the intramolecular hydrogen bonding, and the other is the intermolecular hydrogen bonding. Fourier transform spectroscopy, it is technique that is used to obtain an infrared spectrum of absorption or emission of a solid, liquid, or gas. Here, carry out Fourier transform. It comes from that Fourier transform, that is a mathematical calculation or mathematical process. It is required to convert the raw data into the actual spectrum. You can have a comparison because there are some constructive as well as a destructive interference. This is the typical diagram of a FTIR spectrometer. Now coming to the FTIR interpretation. Here you can see in the spectrum, the range from 4000 to 700 centimeter inverse. The x-axis is the frequency, that is centimeter inverse, and the y-axis is the percentage transmittance. Here you can see this is not the peak, this is the valley or the dip. So these are called as the bands. So from 4000 to 1500 centimeter inverse, that is called as the functional group region, and from 1500 to 700, that is called as the fingerprint region. As we all know that no two persons can have the same fingerprint region. Are are these fingers? They are unique for all the human beings who are present on this universe. Similarly, no two compounds can have the same fingerprint region in the FTIR region, except for the enantiomers, which are non-superimposable mirror images of each other. And the calculation or the interpretation of IR is quite interesting. From here, you can calculate, you can identify, because this is the qualitative as well as the quantitative analysis. So from here you can identify the major functional groups which may be present in the molecule. So here you can see at 1736 a very deep, sharp, intense band, that is of carbonyl group. And this while the interpretation of the IR spectrometer or spectra, we cannot neglect the carbonyl peak if the carbonyl functional group is present in the molecule. It is so intense, so sharp that it cannot be neglected. And the range for carbonyl, it is from 1860 to 1680 centimeter inverse. And you know, carbonyl may be in some other forms also, like it may be acid, it may be ester, it may be amide, it may be acid chloride. And so these it may be, this carbonyl group may also be attached to the aldehyde also and ketones also. So there are various steps that if we have got a carbonyl peak, then we have to go beyond other peaks that which may be present in that. It may be aldehyde, it may be ketone, it may be amide, it may be acid, ester, whatever. And if this carbonyl group is not present there, then again the functional groups may be different. It may be alcohol, primary, secondary, tertiary alcohols will be there, amines will be there, nitro compounds will be there, chlorides or halides will be there. So all the compounds, we have got a chart for this, and the compounds can be identified. They cannot be confirmed on the basis of IR. See, they can be only identified that this particular functional group is present. So identification, structural determination, these are the applications. Study of chemical reactions, study of ketoenol tautomerism, study of complex molecules which may be present, then detection of impurities of a compound, then conformational analysis. All these things can be carried out with the help of FTIR. Then another one is the fluorescence spectroscopy. This spectroscopy is, it's a technique which examines the wavelength of the photons which may be emitted from the atoms or molecules during their transition from an excited state to a lower energy state. It involves using a beam of light. Usually ultraviolet radiation is used that excites the electrons in the molecules of a certain compounds, allows them to emit light of a lower energy, typically but not necessarily visible light. If we go for the devices, fluorometers or fluorimeters are used for a fluorescence spectroscopy. Sorry. Fluorescence, it is measured by exciting the sample at the absorption wavelength, and also called as the excitation wavelength. Measuring the emission at a longer wavelength called as the emission or fluorescence wavelength. This is the instrument for spectrofluorimeter. And the applications of spectroscopy is an emerging diagnostic tool for various medical diseases. It is a non-invasive technique and has been applied successfully for the diagnosis of multi-system cancers with the high sensitivity. It is also used in, among others, biochemical, medical, and chemical research fields. In the biosciences, one of the most frequent applications of fluorescence spectroscopy, it is the high precision quantification. Now I'll give a brief overview of the nuclear magnetic spectroscopy. It is used for the, as the name indicates, it is the game of the nucleus, that is proton. It is a physical phenomenon in which the nuclei in the magnetic field absorb and re-emit electromagnetic radiation, and that electromagnetic radiation is the radio frequency waves. And the energy is at a specific resonance frequency which depends on the strength of the magnetic field and the magnetic properties of the isotopes of the atoms, and it's used to study and research purposes, molecular physics, structure, crystal and non-crystalline material analysis. So principle of nuclear magnetic resonance, it includes the that every nucleus possesses a magnetic moment, and such as hydrogen nucleus or carbon nucleus, it is placed in a strong magnetic field, it will begin to precess like a spinning top. Advanced 1 and 2 dimensional experiments like APT, DEPT, and COSY or HEADCORE, they are use of PFG, are available and necessary tools for the deconvolution of complex mixtures and materials, especially for pharmaceutical characterization. Uh, this is the typical structure of the nuclear magnetic spectrophotometer, and the applications we have, as I have already mentioned, that the structure of the compound can be confirmed only on the basis of NMR, that is how many and what type of protons and carbons are present in a particular molecule. So number of uses are, and the MRI in the medical field, it is based upon this principle. Agriculture, medicine, hospital, functional imaging, in vivo spectroscopy, well logging, NMR microscopy. So a number of these fields are associated with the nuclear magnetic resonance. Now the mass spectroscopy, if I talk about the mass spectroscopy, the structure of the compound, the molecular formula of the compound, it can be, calculated from the mass spectrometry, because there is no light is involved. That is why it is never called as mass spectroscopy. It is always mass spectrometry because it is the measurement of the molecular mass of the compounds. It's a destructive technique, and here actual molecular mass of the compound can be determined. There are various techniques for the ionization, soft ionization techniques as well as the hard ionization techniques are present. This is the diagram of a particular, this is the mass spectrum of a compound. Here you can see the highest concentration peak, abundant peak, is called as the base peak, and the peak that is corresponding to the molecular weight of the compound, that is called as the molecular ion peak. So if I talk about, if I conclude my talk of the day, the spectroscopy techniques or the pharmaceutical analysis, we have got a huge, large, scope in the pharmaceutical sciences, because we can do nothing without these techniques, whether we are from the whichever, whatever department we belong to. But analytical techniques, they have to be monitored and followed properly. So now coming to the in terms of analytical chemistry, the road ahead, the technological advances are being happened in pharmaceutical analysis as like other areas of science. Due to technological advances, more and more pure drug is available and will be available for patients, and drug testing becomes more regulated. The new technological instruments and automation, productivity is dramatically increased, and hence it is benefited the business of the company. Indian pharma companies are more competent to the global market, and with the support of quality analytical data, companies are able to protect their innovations by patenting them. So thank you so much. This is all about my lecture today.
Thank you, Dr. Pooja Chawla, and thank you very much for your kind talk, and thank you for enlightening the analytical techniques in the field of pharmaceutical analysis and pharmaceutical chemistry. And dear listener, we thanks to all for joining us. Please visit our website www.isfcp.org. Thank you, Dr. Pooja. Thank you so much.
Thank you, Dr. Siddharth. Thanks a lot.